Welcome to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Eva Amsen, and experts in the field of cryo-electron microscopy.
Today on Cryo-Talk, we're joined by Mimi Ho, Assistant Professor of Microbiology and Immunology at Columbia University Medical Center. We're talking about her switch between industry and academia.
I hadn't even considered staying in academia, actually, until the summer before I graduated.
Her dog Mighty, who has been visiting the lab.
My students are learning how to mill now on the FibStem, and so he's been going in and hanging out with them while they mill.
And the value of building your own support network.
What I think of as my network is building kind of a support group, right? Like a support network. So I think of it as a support network more than as like a career network, if that makes sense.
All in this episode of Cryo-Talk. Hi, and welcome to Cryo-Talk. I'm Eva Amsen, and I'm here today with Mimi Ho, Assistant Professor of Microbiology and Immunology at Columbia University. Her research group uses cryo-EM methods to study host pathogen interactions in malaria parasites. So hi, Mimi, welcome. How are you today?
Thank you so much. I'm good. Thank you so much for having me.
Great to have you. So I think to start off, why don't you share a little bit about your career so far, and you can tell people how you got to where you are today.
Yeah, happy to. So I guess going back, so I got my undergrad at Berkeley in molecular cell biology, Berkeley MCB, and then after that I worked as a research technician in the lab of Bob Stroud at UCSF, working on membrane proteins. I was a membrane protein biochemist. The lab worked on, X-ray crystallography, using X-ray crystallography to solve structures of membrane proteins. And so I was there for about, I think, 5 or 6 years. And then after that, I worked at Novartis in Emeryville, California, in their in the Novartis Institutes for Biomedical Research in their infectious diseases division for about 3 years. And so at that time was working on kind of both viral and bacterial diseases, different projects. And that kind of really, I think, lit a fire under me. I got really inspired, and kind of that was what inspired me to go back to get the PhD. So then after 3 years at Novartis, I left to go to UCLA to get my PhD with Dr. Hong Zhou. He was my PhD supervisor at UCLA, was there for 4 years and a few months. And then after that, applied for and got a tenure track position at Columbia. And so that's where I am now. I moved here and started my position in January 2020. So it's been about 2.5 years.
Great. It's a strange time to start, isn't it? Early 2020.
Yeah. For sure.
And yeah, you've done both academia and industry. Any interesting differences there?
Yeah, I mean, I think that interesting differences. So I think that in industry it can be a lot more structured. I guess there is a lot more structure, which I really enjoyed, and that's something that I'm trying to kind of bring across in my lab now. So there's a lot of kind of practices that I really liked that in industry, kind of setting intentions at the beginning of the year, having milestones and kind of check-ins every quarter, or like having, the halfway year mark check-ins on different projects and things like that. I felt like worked really well. There's also, they're very, very much into kind of note keeping, like keeping really, really good records. And so that's something that I've also brought across with me into my lab now that I think is really important, especially with the work that we do. Keeping track of all of your data and all of your grids and everything is super important. So
yeah. That's a good habit to have.
Yeah, lots of really good habits from industry that I'm bringing across.
Great. So how did you get started using cryo-EM?
Yeah, so like I said, I was a membrane protein biochemist by training. And so when I started my PhD, I actually fell in love with this protein complex called PTEX. So I was really interested in this protein complex. It's a large membrane protein translocation complex. It's a translocon that works in the parasite that causes malaria, Plasmodium falciparum, and it's responsible for the the transport of all of these hundreds and hundreds of effector proteins that the parasite uses to remodel the host red blood cell. So the parasite lives inside human red blood cells, And in order to be able to do that, it has to completely remodel the inside of the red blood cell. And so it has all these effector proteins that it has to somehow make and then secrete out into the host cell. And so all of those travel through this large membrane protein complex called PTEX. And so I kind of really fell in love with this complex and wanted to solve the structure. And it became obvious, I think, during my rotations at UCLA that cryo-EM was going to be the way to solve this. And so I ended up joining the lab of Dr. Hong Zhou and ended up then using learning cryo-EM from him, and then used it to solve the structure of this large membrane protein complex from malaria parasites.
Cool. Yeah, that kind of brings me to my next question. What makes cryo-EM such a good method to really study host pathogen interactions compared to other things?
Yeah, so I think that, there's two things. So one, I think, was more relevant during my PhD, and one is more relevant now that I've started my own lab. So I think the first thing is that single particle cryo-EM, because it requires so much less material, you need much less protein, and also you don't need the protein to be nearly as homogeneous as other structural biology methods. So for example, like X-ray crystallography or NMR, you need large amounts of large amounts of high protein at high concentrations that are very, very pure. And that's very difficult to do if you're looking at kind of, native or endogenous sources, non-model organisms, right? Mm-hmm. And so I think that one of the major strengths of cryo-EM is that now, because you don't need as much sample, you don't need to be as pure, you can go after protein complexes from non-model organisms, from native sources. So in my case, for example, the protein complex I was interested in, you could not express it in recombinant systems. It just doesn't express properly, it doesn't fold properly, it's a mess. And actually, the organism that I work on, Plasmodium falciparum, actually the vast majority of the proteins are like that. They really don't like to be kind of expressed in recombinant systems. And so that's reflected in kind of the paucity of structural information from this organism. And there's a lot of other organisms out there like that are non-model organisms that, it's just very difficult to express those proteins recombinantly. And so now with cryo-EM, you don't have to anymore. You can actually, there's more and more stories coming out every day of people purifying and solving the structures of these gorgeous protein complexes that they've enriched from endogenous sources. And that is totally something that would not have been possible without single particle cryo-electron microscopy. So I think that's one of the major benefits, especially when it comes to infectious diseases, because we have a lot of non-model organisms in infectious disease that really benefit from single particle. And so now I think that was kind of very much the story of my PhD. And now that I started my own lab, we're taking off in this new direction called in situ cryo-electron tomography, where we actually don't even break open the cell. We're looking directly at our parasites living inside red blood cells intact. And so we just directly take our parasite infected red blood cells, we freeze them, and then we kind of are able to then thin them to a very thin section called the lamella that we can then take tilt series of. And so you can actually see we're getting these really, really gorgeous tomograms of the host pathogen interface. So we're directly able to actually visualize the host pathogen interface at sub-nanometer to near atomic resolutions, right? Either directly or using subtomogram averaging, which is absolutely amazing.
Yeah.
So I think that is also another major strength of cryo-EM, cryo-ET in looking at host pathogen interactions, is that you can actually, with your eyes, look at the interaction.
Yeah, that's such an advantage to really see the actual, the place where it happens, really.
Yes. Yeah, absolutely.
And we already talked a little bit about your career, but has there ever been a moment, either in your research or in your career path, where things took a surprising turn?
Yes, definitely. So as you may have seen from kind of the first question, my path to where I am right now has been kind of pretty convoluted, not straightforward, right? So I went, I was in academia for a while, then I went to industry, went back to academia, and actually my original plan, so the original reason to go back and get the PhD was actually so that I could go back to industry. So I was having so much fun at Novartis, working with these amazing people on these really exciting projects that really had, immediate downstream effects, like downstream applications. So one of the projects that I was working on the drug reached phase 1 clinical trials by the time that I left the company,
Wow.
which was really exciting, right?
Yeah.
And so my original motivation for going to get the PhD was, to get the PhD, because I could see that, in order to be able to kind of participate and kind of, gain more influence over projects and just kind of, give more, be more involved at a higher level in industry, I needed that PhD. And so that was my original motivation of taking a huge pay cut and going back to grad school to get that piece of paper so that I could go back into industry and do really exciting things. And so that was kind of my game plan during my PhD. And I hadn't even considered staying in academia, actually, until the summer before I graduated. I didn't know at the time that I was going to be graduating so soon. So I had this PTEX, I had finally solved the structure, and we had, kind of gotten the paper accepted. And so I was presenting it at a conference at a GRC. It was a host parasite interactions conference. And when I was presenting it, a lot of PIs came up to me afterwards and they were like, that's awesome. Like, what are you planning on doing next with it? And I was, what would you want to do next? And I told them what I wanted to do next. And then they were like, so are you applying for positions? Like what's happening? And then I was like, oh no, I'm going back to industry. And everybody was just like, but you have all these plans, you have all these things that you want to do with this project. Like, why wouldn't you even consider the possibility of staying in academia and maybe pursuing some of these exciting new avenues? And so this was something that I had these blinders on and I wasn't thinking, right? And so they, two of the junior PIs at the conference, who are very close friends of mine now, spent kind of 2 hours on the last day of the conference just sitting there and talking to me and convincing me that I should at least give it a shot. And so, I kind of had a bit of a meltdown in Boston Logan Airport on the way home, because it was just, it was something that I had not even considered. And so I was freaking out a little bit. I called my sister and she was like, okay, so I think the reason you're freaking out is because you're trying to make this decision right now, this huge decision about whether to stay in academia or go back to industry without any information, right? Like I was trying to make the call right there in the airport.
Yeah, you don't have to do that.
Yeah, and so she was like, why don't you just apply to both positions and then go on the interviews, you'll learn more and then you'll feel better about making the decision because you have more information. So that's what I ended up doing. I applied for some independent fellows positions. I also applied for just, some straight up faculty, tenure track faculty positions, and I applied for industry positions as well. And then I ended up going on all of the interviews, and in the end, I chose the faculty position because it was the position that allowed me to do all of the things that I loved. So I kind of really fell in love with this parasite. It's amazing. It does this really cool, all of this really cool biology that you don't see anywhere else. It's really unique. And obviously, I also very much love cryo-EM and cryo-ET. And so this was the position that allowed me to kind of continue to be able to do all of those, if that makes sense. So yeah, I completely unexpected. I was totally not planning to end up in academia, but here I am.
That's a great story. And I can totally imagine how a room of academics reacts when you say that you actually want to stay in industry. They must have been very shocked.
Yeah. I think attitudes are changing these days. I think definitely in the old days, people were very hardcore academia, but I think people are recognizing these days that, you can do amazing science in either setting. There's amazing science happening in both locations. I mean, I think like what has happened with this pandemic and the development of this vaccine has been a really beautiful example of the fact that you need contributions both from academia and industry in order to get something big like that to happen, right?
Yeah.
So I think more and more people are recognizing that it's not one or the other. A lot of times it's kind of a really cool interplay between the two.
So what's next? What are you looking forward to in your researcher career?
Yeah, so we're currently pushing to try to get our first paper out as a lab. So that's one thing that we're really looking forward to. I just had my first 2 graduate students join a couple months ago, and they're amazing. So I'm really enjoying watching them learn and kind of fall in love with cryo-EM. I think that, it wasn't that long ago for me, the first time that I fell in love with cryo-electron microscopy, but it's really fun to watch people kind of go through the first steps where they, I don't know, all of that joy is really, really fun. So that's something that I'm really enjoying right now. And like I said, we're pushing really hard on this paper right now. So we're really looking forward to getting that. And then I think
that's something to look forward to.
yeah, definitely. And then I think from then we've been over the past 2 years, we've spent really kind of from scratch, like teaching ourselves how to do the cryo-ET, because we didn't know how to do any of it at the beginning, like, you know, how to prepare the cells, how to do the, you know, freezing, the milling, the data collection, or any of the data processing. And we started literally from scratch and have been kind of teaching ourselves. And so I think after this first big paper gets out, this is kind of our paper to say, like, we can do this, like, you know, we're here. And then after that, we're really, really excited to kind of push the boundaries and see, you know, what new things we can discover in terms of how this awesome parasite interacts with its host using this new method. So very excited.
Cool. And you've also been working on a podcast. Now we heard a little bit about that podcast 2 episodes ago when we spoke with Liz Kellogg. And then the episode after that, we spoke to Mike Tamfrocco, who we didn't talk about the podcast in that episode, but you're all in this together, I believe.
Yes.
So tell us a bit more and what's happening now.
Yeah, no, definitely. So the podcast was kind of inspired by we formed this kind of network during the pandemic. That's one awesome thing that's come out of the pandemic is there's a lot of networks that have formed, like support groups almost, that I don't think would have happened without the pandemic. And so through that, Liz and Mike and I got really close. And so we were kind of hanging out. We have these kind of really awesome conversations, like really broad ranging conversations about all kinds of things. So not just specifically talking shop, but also talking in terms of like, kind of what it's like to start up a new lab, especially in the middle of the pandemic, and kind of, you know, career paths and kind of plans and things like that. And then also kind of our hopes and dreams for the future for cryo-EM. And so out of all of these kind of really wonderful conversations, we thought, well, you know, why not share this with people? So we thought it would be really cool. There's obviously, you know, these interviews are great, but we wanted to go for, there's obviously lots of people already like you already doing these amazing interviews. So we wanted to go for something a little bit different. And so what we did is we have this series of 6 interviews where it's a little bit like fireside chats, a little bit almost. So it's the 3 of us and we're interviewing somebody. We invited a few different people in the field, kind of leaders in the field, and also people who are brand new, just starting out in the field. And we tried to make sure we had covered a broad diversity in terms of kind of different career paths. And so we've invited these guests to come, kind of they're each one, I think maybe 30 minutes, 20 to 30 minute episodes. And we really tailored the questions to the person. It was really special because each of the people that we invited were people that we personally knew and had relationships with. And so they were kind of really awesome interviews because they shared a little bit about their career path, how they got to where they're going, which I think is really great. A little bit about their vision for the future, and then also kind of, you know, more about their individual science as well. So I think it's a little bit different than, you know, the normal kind of interview. And so I think people, I mean, we thought it was really fun to do. And then afterwards we've been watching, like we just watched the last one. So they've been kind of doing post-production and then sending us the kind of final pieces for us to kind of go in and make the last minute changes that we want. And they're amazing. Like they're absolutely, cool. All of them turned out so well. We're really excited to share it with everybody. And we were planning on doing it as a one-off, but I mean, if everybody else thinks these are as cool as we do, we might have to do a second season. So.
Yeah, and by the time that this episode goes out, your podcast should be live, so we can put a link in the description as well.
That would be amazing. Yeah, I mean, it's literally like there was just I want to say, you know, all of the episodes are my favorite. Actually, they're all from completely different perspectives. Some with really senior people, kind of very forward thinking. The one with Ifan where he talked a lot about kind of mentorship and how to build a healthy lab environment and how to kind of just, you know, foster like a broader, like healthy, you know, just a healthy collegial community. Just a lot of really amazing stuff in there. So I'm very excited to share it with everyone.
Yeah. And I know from experience, it's fun to interview people for a podcast.
It is.
I'm sure you had a lot of fun. Yeah.
Yeah, no, we had a ton of fun with it. Very much so.
So it sounds like this was already fun, but what else do you do for fun?
Yeah, so I guess I read a lot. I like reading. sometimes if I want to just chill, then I watch TV also. But I think just to kind of calm down and relax, I like hanging out with my dog. This is Mighty.
Aw.
Yes. So he's a Chiweenie. He's about 13 years old now, I think.
Oh,
He's
that's
been
so
with me a
old.
long time. And so yeah, we like going on walks with him. There's a bakery that we really like that's down on the Upper West Side, and so every weekend we walk down with him.
Aw.
So, yeah,
That's
I
great.
spend a lot of time just hanging out with him.
Yeah, that's fun. Especially going for walks. I just have a cat, so I can't really go for walks with my cat.
Yeah.
He's just sleeping most of the time.
Yeah, I mean, lately, so I've been taking him in, and actually, with my students are learning how to mill now, on the FibSem, and so he's been going in and hanging out with them while they mill, and yeah, they've really enjoyed that. I think they've gotten to the point where they refuse to mill without him, actually.
It's part of the team.
Yes.
I got a couple of quick fire questions for you as well. And we asked
sure.
everyone the same questions so that we can kind of compare, like, what are all the cryo-EM researchers doing? So, do you prefer the city or the countryside?
I like them both. So I grew up in Ames, Iowa, which is definitely, I think, what most people in New York would consider the countryside. And it was an amazing place to grow up as a kid. So I spent kind of all my time running around outside, building forts, like raising baby birds, all kinds of stuff, right? And then now I think as an adult, I prefer to live in a city. I think I'm definitely a big city person, but it's really nice to visit the countryside to decompress.
Yeah, it's a good balance. And do you like to cook?
I do like cooking. I love cooking. I like baking even more, actually. So I started baking before I started cooking, but yeah, I do really like to cook when I have time.
What's your favorite thing to make?
I don't have a favorite. I really like miso salad, so I make a pretty good miso salad. There's also a lot of kind of just I don't know, basic home food like Chinese dishes that I learned from my mom that I like making. There's this Chinese spare ribs with green beans that I really like as well.
And you mentioned that you like to read. Is there any books that you'd recommend?
Oh, you should have warned me about this one. I have so many.
Or several.
Yes, I have a ton. So I really like Ender's Game. That's like my go-to that I always recommend to people is Ender's Game. It's a science fiction one. I also really like War for the Oaks, which is more of like an urban fantasy. I like Seven Eves actually as well. The author is from my hometown actually, which I didn't discover until I was already like really into his books. But yeah, I could give you a whole list, but I think we probably don't have time.
No. And what about TV? Do you watch TV or films?
I do. Both. I watch more TV these days than films. Although I have to say that I went and watched the new Top Gun twice in theaters. I'm a huge Top Gun fan. Like my guilty pleasure. Like I absolutely love Top Gun, and so we went and watched it in theaters twice actually, and it was equally good both times. The new one is really good. If you're a fan.
It's always better in the cinema, especially those big
that one
films.
Particularly, yeah, I think it's worth watching on the big screen. Just because the visuals are amazing, and so it just won't have the same effect if you don't watch it on the big screen.
And do you like listening to music?
I do.
And what kind of music do you like?
It depends on what I'm doing. So if I'm writing, I like listen to lo-fi music, so it's not there's no lyrics. Like I can't when I'm doing research or when I'm doing work when I'm working in the lab, I have a hard time focusing if I listen to things that have lyrics, because then you like start singing along and then you like lose your place. So the lo-fi music works really well for that because there's not any lyrics to get caught up in and it's pretty chill. But it keeps you awake as well. So that I really like.
And there's all these lo-fi playlists now as well.
There's some really great ones. Yeah, I discovered them. I don't remember when I discovered them, but yeah, they're really good for all kinds of things, for picking particles when you're doing something that, you have to stay awake for, that like you just kind of have to power through. It's really great for that too. So gotten really into that recently.
So I know you love science, but if you had to do something else, if you were not a scientist, what would you be?
It would probably be something in the food industry. I'm a huge foodie. I love food of all kinds. And so I think if I hadn't already done it, I would say that I would want to be a bartender, because I think that's a really cool job. You get to like interface with a lot of people. It is kind of food centric as well. But I did bar back for a couple of months already when I was in between jobs, right after I graduated from undergrad. And so now I think it would be fun to own like a bakery / coffee shop, cafe type thing, maybe.
Yeah.
I don't know that I would want to do like a full-on restaurant, but like
I think that would be mine as well, like running a little cafe.
Yeah. I think that would be amazing. Maybe on the beach somewhere in a beach town.
Yeah. And it's funny that you said bartending, because in my mind that is so much like a lab work. It's like mixing things very much... and keeping things clean and fast paced and
no, definitely. I mean, I think that's why also I love cooking and baking, because it's basically like doing biochemistry, right? Like you're mixing things.
So finally, I got one more question that I ask everyone, and that's, do you have any advice for researchers who are just starting out? So maybe thinking about your new students, what would you advise them?
Yeah, so I think that the first thing that I advise them is to keep an open mind. Don't put on the blinders. So that, obviously, I mean, I think from my career path, it's pretty obvious that like, you have to grab opportunities when they come up, and they might not always come up when you expect them to. They might not always be. A lot of times they won't be what you're expecting. So just keep an open mind and keep your eyes open, and so that you don't miss those opportunities when they come along, because a lot of times the unexpected ones are actually the best ones. So I think that would be my first piece of advice. My second piece of advice would be to find good mentors, and by that I don't just mean your PhD supervisor. I mean like at all levels, right? So I think that it's really important, if you're a graduate student, for example, to kind of find a couple, like 1 or 2 postdocs and 1 or 2 kind of maybe people that are at the assistant professor level, and then 1 or 2 people that are mid-career or senior, like people at all different levels, because they're all going to give you different advice, right? Because they're at different life stages, I guess. I was about to say life cycle stages, because my mind is always thinking about parasites. But yeah, they're all, we're all at different stages. And so the advice that they have to offer is very different. Their perspectives are very different. So I think it's really important to find mentors at all different levels. And then also obviously at the same level as you. I think when I was younger, whenever people said networking, I was always like, oh, like networking, it feels kind of artificial, and like you're putting yourself out there, and it, there's ulterior motives, but actually, so I don't like that. But I do think that, these days, what I think of as my network is building kind of a support group, right? Like a support network. So I think of it as a support network more than as like a career network, if that makes sense. And so that's why I'm saying, like during the pandemic, I formed kind of a support network with my parasitology colleagues. So there were a few other junior PIs who all started around the same time as me in parasitology, and we formed kind of like a support group. Almost where we have a text chain, and we like share kind of all of our, whenever we're really sad about something or really excited about something, we have somebody who are like exactly at the same stage as you, and so they get excited. They know exactly where you are.
Yeah,
right? And I have the same thing in cryo-EM. Yeah, so I have a group of that in parasitology, and then one also in cryo-EM, my 2 fields. And so I think that's super important. They're not. I don't think they're my friends. I don't think of them as like right. But it's I think that's really important, is to build a support network of peers that understand what you're going through and are in it with you, who can kind of really share your wins and your losses with you.
That's great advice, and I think it's really useful also for people to know, that word, like you said, networking sounds boring and horrible, but it really is just socializing and talking to people.
Yeah, no, and I think definitely thinking of it as a support network helps a lot.
Great. Well, thank you so much. That brings us to the end of our episode today. So thanks again for coming on the episode, and thank you everyone for listening to or watching Cryo-Talk.
Thank you so much for having me. I had a really good time.
Thank you for listening to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/cryotalk.